Okay, so this is video 4 of Unit 4, Sensation and Perception. What we will review is how light actually enters the eye, is processed in the eye. We'll also go over the theories of vision. Information on here can be found on page 128 to 133 in your textbook. So let's review this image. Hopefully you have it somewhere in your notes, and also a handout was given in class on the parts of the eye. We'll start right here with the cornea. Okay, cornea, remember, is the clear plastic covering of the eye that protects the eye. Okay, that enters the cornea. Okay, and then goes through the pupil. The pupil is the black area of the eye that light actually passes through. What controls the amount of light entering the eye would be the iris, which is the muscle portion of the eye, that it's either going to dilate or constrict. Once it goes through the iris, then the lens is going to use accommodation, which helps focus the image on the retina. Okay, and the retina is the multi-layer area of the eye, which is made up of cones and rods, which help us detect color and black and white. Also, transduction takes place here. The sharpest area of the retina is known as the fovea, which the majority of the rods and cones are found in the eye. This helps us understand sharpest vision. And last but not least, it then exits the eye through this is supposed to be here, it's supposed to be the optic nerve. Okay, so you might want to change that here if we put optic nerve. The optic nerve, remember, are made up of ganglion cells that help send the message to the brain. Okay, the blind spot is the area where this connects, the optic nerve connects to the brain, connects to the retina, and this is where vision can't be processed. Vitreous gel is what the eye is made up of. We don't need to know this, but it was included on your diagram. So again, this is a good area to jot down the visual processing. It goes from the cornea to the pupil to the iris, which will either expand or constrict. The lens helps focus it on the retina. The retina is the where the eye transduces the characteristics of light into neural signals that the brain can process. This is transduction. It happens in the retina and the rods and cones. It then goes to the bipolar cells, to the ganglion cells, to the optic nerve, all the way to the thalamus, and then to the visual cortex. So again, make sure you have this memorized, written down in your notes. Once it gets to the brain, it then enters what's known as the thalamus, and we've talked about this. It receives all the sensory information except smell. It's known as the switchboard of the brain. So it goes to the two areas of the thalamus. Okay, once it leaves the eye, goes to the one area of the thalamus and the other area, and then the thalamus sends it to the visual cortex, which is known also as the occipital lobe. Vision is processed here in the brain. This is where perception takes place. We've been talking a lot about sensation, picking up. Perception takes place here, where it interprets what we see. So what allows us to understand what we see is what's known as feature detection cells. These are found in the visual cortex, the occipital lobe, and it's our ability to understand a scene's specific features. It helps us understand pictures as a whole, okay, particular edges, lines, and angles. We're able to see all the details on this squirrel, okay, because of our feature detection cells. It allows us to understand that the squirrel's tail is ending here, and the background is now, we're understanding the background of images, lines of things, angles. All of this helps us understand the picture as a whole. Okay, damage to certain areas would be damaging processing different components and understanding when things start and finish. We also have parallel processing that takes place in the brain. Okay, our brains are able to process many things in a scene at once, simultaneously processing several aspects of a problem. We're able to understand color of images, motions of things moving, the form of the bird, and how far something is, the depth of things. Okay, this wing obviously is closer to this wing because we understand the component of depth. Okay, we're able to understand motion of things because of parallel processing. All of this is happening at once. So imagine you understand that water flows out of a faucet, and you could see the different parts of the water, the different raindrops, you could see it moving, you could see the color all at once. However, if this was damaged, you wouldn't understand a faucet running. So there was a stroke patient that had areas of the brain that control processing damaged, and they didn't understand when you were pouring a cup of tea that it didn't look frozen. Okay, so somebody who didn't have these components would see things as a still image or wouldn't understand movement of things. There are two theories, okay, the Young-Helmholtz theory and the opponent process theory. You need to know both of them. This helps us understand colors. So the first one is known as the Young-Helmholtz, or it's known as the trichromatic theory, two different names. Okay, it was developed in the 19th century by Hermann von Helmholtz and Thomas Young, hence Young-Helmholtz theory. Okay, what they believed that the retina contains three different colors. We have red, green, and blue in our photoreceptors, our cones. So we have components that allow us to see red, green, and blue. The combination of these allows us to understand the different components of color or light. Okay, so if you blend, this is if you've taken an art class, you blend red and green together. Okay, for light, it would make up yellow. If you blended red and blue, it would make up a fuchsia purple. If you blend blue and green, it would help make like a teal. And if you blend all the colors, okay, this is light, would actually make up white. Okay, this occurs in the visual cortex. If you are colorblind, you may lack a certain cone, whether you're lacking red or you're lacking green or you're lacking blue, which wouldn't allow you to see certain colors. It's either if you're lacking two, you're dichromatic, and if you're lacking one, it's monochromatic. Okay. The opponent process theory is a little bit different. In 1878, Ewald Hering stated that some receptors may be turned on or off by certain receptors in our eyes, which creates what's known as the opponent process theory. The opponent process theories are red, green, yellow, blue, and black and white. And this occurs in the thalamus in the visual cortex. So what they're saying is are certain cones that control all, let's say that control us detecting red. If we soak up all of the white in our eyes, what it can turn on is it can turn on then the black photoreceptors for them to be seen. If we turn on, if we soak up all our eyes with red, what it's going to oppose is the green color. And if we soak up all the yellow, it's going to oppose the blue color. So this is helpful to remember. You need to know what are the opponent process pairs. Red, green, you may think of holidays. Blue and yellow, you may think of light and color, and then black and white is yin yang. So take a minute and I want you to see how the opponent process theory works. Okay, it often creates what's known as after image. After tiring your neural impulses to seeing a ton of black, you're going to see white. If you see a ton of green, you're going to oppose red. If you see a ton of blue, you're going to oppose yellow. Okay, so these are the opposing images that create after images. So stare at this picture for a little bit of time. I want you to stare right here between the yellow and green and black area, right in the center. Pause it for 30 seconds and then hit play. And what you should see then is the American flag, because you've soaked up all of the you've soaked up all of the yellow, which is going to oppose blue. You've soaked up all of this green, which is going to oppose red. You've soaked up all of this black, which is going to oppose white. And this is known as the opponent process theory. Sometimes you do have problems with your vision. I do like to talk about this. There's two different types. You may have nearsighted or farsighted. Nearsighted is called myopia, which is the degree of nearsightedness affects your ability to focus on distant objects. So when you're nearsighted, you have a hard time focusing on far objects. Nearsighted may develop gradually or rapidly and worsening during childhood and adolescence. Nearsighted tends to run in family. So nearsighted, you actually have a hard time seeing in the distance. Okay. Farsighted, on the other hand, is hyperopia, which is common vision conditions in which you have problems seeing, you see the distance very clear, but what happens is you can't see close up. Farsighted usually is present at birth and tends to run in families. Usually it's easy to correct with eyeglasses and contact lenses. Another option a lot of people get is surgery. Okay, so farsighted is where you have trouble seeing up close, and nearsighted is where you have problems seeing far away, which you think would be the flip or the opposite. Okay, so make sure that you go